Alright, so in week 9, we're going to be talking about the basal ganglia. And we talked about the basal ganglia some last bi-term, but we're going to be looking at it a lot more in detail this week. So let's go back to where we were. So this picture should look familiar. However, we're going to add a little bit of complexity to it. So is that relatively even? Yeah. Now, I normally don't draw it separately like this, but because we're getting more into the weeds of it, let's get more into it. And I'm just going to draw it first, and then we'll be good. But this would obviously be a frontal cross-section of the brain and some of your brainstem. So this is your cerebrum, and then this is your midbrain. So let's go ahead and draw our ventricles. So we have the third ventricle, we have the lateral ventricles right here. The thalamus is going to sit in here. And then now we're going to start drawing our basal ganglia. So this is going to be our caudate nucleus. And then we will draw our lentiform nucleus. And then we'll split our lentiform nucleus up into its divisions, and then we'll draw all the subthalamic nuclei. Now, if we look back to, and then this is our substantia nigra right here, and it's divided up into two parts as well. But if we go back, so we have the caudate nucleus, so I'm going to abbreviate these, so the caudate nucleus, and then this part right here is the putamen. And then we have the globus pallidus, but I drew little splitting lines through the globus pallidus, and that's because we have a globus pallidus externus, and then more internal, you have the globus pallidus internus. But this whole thing right here, if we don't include the putamen, is the globus pallidus. Now, it's at this point where we can start to add different structures and we can get different names. If we combine the caudate nucleus and the putamen, then we are going to form what is called the striatum or striatum. I'm not sure. And then if we take this whole piece right here, so the putamen and the globus pallidus, both parts of it, then we get what is called the lentiform nucleus. This little guy right here is going to be your subthalamic nucleus, so we'll abbreviate that SN. And then this is your substantia nigra. But it has two different parts as well, noted by the little dashed line there. So this top part is the substantia nigra pars compacta, and then the bottom part is the substantia nigra pars reticulata. So compacta and reticulata. So whenever we were at immersion and we were talking about, those dopamine neurons that were involved in the basal ganglia, and we said, oh, that's the substantia nigra, we never clarified which part. And what we will now. That part that has the dopamine neurons is the substantia nigra compacta. The reticulata is involved more in your indirect pathway. But now that we've mentioned pathways, let's go ahead and go into that. So let's erase these names here. Hopefully you got those down. You can always just rewind. All right. So how we'll do it is we will split this up into two cross sections. So I'll have the direct pathway on this side, and then we'll have the indirect on this side. All right. So when it comes to our direct and indirect pathway, a good place to start is what do each of them do? So if we remember, the basal ganglia is responsible for our movement plans. It is responsible for starting, stopping, or modulating a movement plan. The direct pathway is going to be more responsible for starting the movement. more so initiating the movement. Whereas the indirect pathway is going to be more stopping movement. So if movement is not happening, but we need it to be, then the direct pathway is going to fire. Whereas if we are moving and we need it to stop, then that would be the indirect pathway. All right, so let's start here. So real quick, just to make sure this makes sense, when I draw a neuron in green, so we have the cell body and the dendrites here, that would be here, and then the axon and then the axon terminal. If it is green, then what I am getting at is this is a excitatory an excitatory neuron, and most of the time, at least for our purposes with the basal ganglia, it is going to release. The reason why it is excitatory is because it releases the neurotransmitter glutamate. So glutamate is whenever it travels across the synapse, the other cell is going to be stimulated, which makes this an excitatory neuron. Versus if I draw it in red, this would be, what I'm getting at here is this is an inhibitory neuron. And why is that? And that's because it's going to release the neurotransmitter GABA. Now, are there other neurotransmitters? Yes. But when it comes to the basal ganglia, those are the main two. And then we have a third type, which is excitatory or inhibitory, depending on what exactly we're talking about. And we'll do that one in blue. And this neurotransmitter is dopamine. So this is going to be the ones that are coming from the pars compacta of the substantia nigra, or excitatory. If I can spell inhibitory. Okay. All right, so let's start with the direct pathway. So this is more of our starting movement. Well, we know that it has to come from the cerebral cortex. So let's go ahead and draw that. So this is going to be the start of our neuron, because remember, the outside of the cerebral hemispheres is gray matter. So that's where our neuronal cell bodies are going to live. And I'm going to draw this as two different axons. Just know that it's not actually like that. But it makes the drawing a lot simpler. All right, so from the cerebral cortex, we are going to go into the striatum. But the striatum has two parts, right? The caudate nucleus, and specifically, we're going to be looking more at the head and early tail of the caudate nucleus, because the rest of the tail does other functions. But think more head, early tail for the caudate nucleus. And then it's going to also go to the putamen, or the putamen. And then, so that is going to be a excitatory neuron. So it's going to release glutamate. And then it's going to synapse on a inhibitory neuron. And then it's going to go to the globus pallidus internus, so the more internal part of the globus pallidus. It's going to synapse with another inhibitory neuron, and then it's going to go to the thalamus. But you're like, okay, there's like a million different nuclei in the thalamus. Which ones is it? It is going to be the ventral anterior and the ventral lateral nuclei of the thalamus. And I'm pretty sure it's more the ventral anterior. However, all of the resources I've read say that it's the ventral anterior and so VA and VL nuclei of the thalamus. And then the thalamus is going to be a excitatory axon, and it's going to go all the way back up to the cerebral cortex. So this is the pathway, but how exactly does it work? Because as of right now, we're just looking at a bunch of lines, right? So if we look, so this is an excitatory neurotransmitter, and since it is firing, that means we're increasing this amount. Oh, I should also mention, so this is also going to the pars reticulata, where it is also synapsing on an inhibitory neuron, and then it goes to the thalamus as well. But this one doesn't seem as important as this one, but just keep in mind it's practically the same thing. But I figured I'd go ahead and write that down. But yeah, so this is increased. But we look at this and we go, well, we're releasing glutamate, which means that we're going to increase these neurons firing. So we go, okay, so these increase. So let's draw little arrows. Okay, and if we increase all of those, then we're going to release GABA, which is going to inhibit the next neuron that it goes to. So since we are inhibiting an inhibitor, well, since we are exciting an inhibitory neuron, then we are going to increase the amount of inhibition. So there we go. So we're drawing little down arrows. All right, so there is a very small amount of this neuron firing, which means that since this is an inhibitory neuron, that it's not going to send a very strong inhibitory signal to this neuron here, which means that this GABA right here isn't going to have much of an effect, which means that this neuron from the thalamus to the cerebral cortex is heightened. It's going to be more stimulated. And that's going to come down to a big thing. If you are inhibiting an inhibitor, then you are stimulating whatever that's targeting. All right, so now for the indirect pathway. So, like I said, the indirect pathway is going to be more responsible for stopping movement. So if movement is already happening, then this is going to be more stopping it. So we're going to start, let me grab a different one here. So we're going to start at the cerebral cortex. We are then going to the striatum. And then this one does something a little weird too. It's going to go to the subthalamic nuclei and also stimulate it. But let's stick with what exactly it does to the striatum first. So at the striatum, it is going to synapse with inhibitory neurons. Remember, those are the ones that are going to secrete things like GABA. And then it's going to be sent to the globus pallidus externus instead of the internus. It's also going to send a message to the pars reticulata. And then those things are going to send a message of inhibition to the subthalamic nuclei. But the subthalamic nuclei is going to release a stimulatory neuron to the globus pallidus internus. And then the globus pallidus internus, it sends its neuron, and then this is where it should start to look familiar. The globus pallidus internus does inhibition to the thalamus, but the thalamus does stimulatory information to the cerebral cortex. So that's our pathway, but where exactly does that leave us? So let's go through it. So we know that since these are excitatory neurons, because they release glutamate, which is an excitatory neurotransmitter, all of these are going to stimulate things, noted by the arrow. That's why the arrow's up. Okay, so we end up stimulating this neuron right here. So this one's stimulated as well, and then this is also stimulated. Okay, and then, what does it do to those neurons? So to the subthalamic nuclei, since we are inhibiting an inhibitor, this neuron right here isn't going to fire very much. And then since we're also inhibiting an inhibitor, this one also isn't going to fire very much. So look and see what we've done to the subthalamic nuclei. And this is key for the indirect pathway. What we've done to the subthalamic nuclei is that we've not only freed it from inhibition from the globus pallidus externus and the substantia nigra pars reticulata, but we're also stimulating it. So this guy is being way stimulated, which means that if we stimulate, if this neuron is very stimulated, then that message is going to go through the other one. And since this neuron does very strong inhibition, and we stimulated it very strongly, then when it releases GABA, then it's going to make this neuron fire very slowly. And if this one's not firing very slowly, not much movement is going to be produced, which is why the indirect pathway is involved more with stopping movement, whereas the direct is more with starting. All right, so it's not the easiest thing in the world, but that's where we are. So now let's talk about so the basal ganglia, we've said it starts, stops, and modulates movement. But we've really only talked about starting and stopping movement via the direct and indirect pathway. This is where it comes down to, well, what does the substantia nigra pars compacta do? And we're going to note that with these blue markers. And remember, it's blue because it's going to release dopamine. So dopamine, so what it's going to do, whether it is excitatory or inhibitory, depends on what neuron it lands on. So what kind of receptors the receiving cell has. So in the direct pathway, these neurons have what is called D1 receptors. So when dopamine comes on here, it's going to bind to those D1 receptors, and that is going to stimulate those neurons. And since we are stimulating an inhibitor, then what are we going to do? We're going to end up stimulating this neuron even more, which means that we're going to end up, so it also stimulates this one as well, which means if that one's, which means if we stimulate this one, it's going to inhibit this even more. So if we're inhibiting even more, then we're going to end up stimulating that movement more and more. So it's going to stimulate the direct pathway even more than it already was. But what does it do in the indirect pathway? So still we're talking about the pars compacta. So we're coming up to these neurons right here. Now, does it also go to the right up here? Yes, it does, because it goes to the whole striatum. But in the indirect pathway, it is going to bind to D2 receptors. And you're like, okay, well, what's the big deal? D2 receptors, when they get stimulated, whenever they bind to dopamine, it is going to actually inhibit those neurons. So we're going to end up fighting against this pathway. So it inhibits this one. And if we stimulate an inhibitor, then we're going to end up increasing. And what you could almost do with the indirect pathway, just do the opposite of whatever it was doing. So we're going to go, so this is up, so it's going to go down. And then this was up, so now it's going to go down. Well, this was down, so it's going to go up. And then look what we did here. This is going to end up being less stimulated, which means that there's going to be less of an inhibitory response, which means that the neuron from the thalamus to the cerebral cortex is going to be less inhibited. So what does that end up doing? That ends up wanting us to move more. So the substantia nigra pars compacta, those dopamine neurons, it's going to be more geared to starting movement, and that's because it helps the direct pathway fire better, and it inhibits the indirect pathway to make it fire less. So so yeah, it's all about starting that movement. See, is there anything else? All right, no, that's about it.